Students, today we are going to study the rest part of the cell junctions. Cell junctions, as I mentioned in the last lecture, that they are meant for cell-to-cell interconnections and they integrate the cellular activity, which is the mark of a tissue. If the activity of the individual cells they aren't integrated, that means the cell is and the cells are aggregation and not cannot be designated as tissues. And in the last lecture, I had mentioned tight junctions, the type of cell junctions. The tight junctions are also called as zonally occludents. They are meant for prevention of passage of fluid from the lateral sides of the two cells. That means they are involved in sealing the lateral passages which are present in between the two cells. Then the second kind of junctions which I had mentioned were desmosomes, and they are meant for provision of strength and rigidity to the tissues. They hold the cells securely together, and they are not involved in prevention of passage of fluid. Rather, they give the tissue some strength. They are spot desmosome, one of the kind of desmosomes, also called as macular adherens. They are also called as spot welts, and they are button-like points of contact in the central stratum between plasma membrane of adjacent cell. They are proteinaceous in nature, as indicated that when they are subjected to trypsin, they get digested. The central stratum between the two membranes consists of specific desmosomal proteins called as cadherins. They bind the cells together. You just see this diagram. Beneath the plasma membrane is a disc-shaped plaque of around 50 to 20 nanometer in thickness. Some filaments, which are also called as intermediate filaments or tonofilaments, which acts as cytoskeletal elements, they get out of these plaques. You can see in the diagram that the plaques are depicted in yellow color, whereas in gray colored you see the tonofilaments, which are intermediate filaments, and on both sides there is cytoplasm of cell 1 and cell 2, plasma membrane of the two cells, and in between the two cells you can see the cadherins, which are spot desmosomes, maculae adherens. You can see in the diagram. They are just holding the two cells together but cannot prevent the lateral passages of the solute molecules. Another kind of desmosomes are belt desmosomes. They are also called as zonula adherens. They forms belt that surrounds each cell, causing cell to cell adhesion, and they are located below the tight junctions. You can see the diagram of these belt junctions. They are specifically conspicuous in columnar epithelium of intestine, and they are present and surrounds the cell like a belt. They have actin filaments arising out of catenins, and both the cells you can see, and in between the two cells you can see the belt desmosomes holding the two cells like belt. Then another kind of junction is gap junction, or gap junctions, which are very important for the cellular integration and metabolic integration of the surrounding cells. Because a chemical stimulus, if received by a few cells, can be transmitted to other cells through these kinds of junctions only. Also, the nutrients, metabolites, and other solutes, enzymes, they pass through these gap junctions. So, gap junctions are very, very important for the metabolic integration of the cells. It is a very important evolutionary event having evolutionary significance. The gap junctions are actually the specialized channels of around 2 to 3 nanometers, and communication between the cell and exchanges of small molecules of the diameter in and around 2 nanometer diameter for the purpose of metabolic integration, as I mentioned, for metabolic coupling or metabolic cooperation, these gap junctions are very important. Because all the cells, they cannot synthesize all the important ingredients of the cellular machinery like CAMP, ATP. They need to be transported to other cells, and for their passage, gap junctions are involved. You can see the diagram of these gap junctions. The metabolic events or reactions, they are initiated in one cell, and for example, we can take pancreatic SNS. One cell, if it is stimulated by hormone, the excitation is passed on to other cells also by opening of certain channels like calcium channels and other channels, and this is how cellular metabolic machinery is integrated. This is the diagram of gap junctions. They are also called as connections, and you can see in the middle part that there is a small passage of around 2-3 nanometer diameter, from which the molecules and other metabolites can pass. So students, this is all in this lecture. Thank you very much.